Here in part 2, let's talk about angiosperms and why they're more efficient in terms of transporting water, and why this coincides with a decrease in atmospheric CO2 concentration. So when did the angiosperms first begin to expand? It was at the end of the Jurassic, and at that time we'd had Pangaea, the supercontinent, break up before, some 225 million years ago. Gondwanaland and Laurasia emerged from that. Both of these continental distributions affected the circulation of water and energy throughout the world. When the Jurassic world was ending, 150 million years ago, we began to see the breakup of the continents into the continents that we begin to recognize today: North America, South America, Europe, India, Australia, Antarctica, and Asia. As they broke up, the ocean circulation changed. That made the continents become drier. So if you look at this graphic here, we have time on the bottom axis, unlabeled, and we see a time when biodiversity was dominated by conifers, but beginning 145 million years ago, we find radiation, expansion in the number of species of angiosperms, so that angiosperms came to dominate our planet. Coincident with this is a decrease in atmospheric CO2. This becomes important because, of course, carbon dioxide is required for photosynthesis, and carbon dioxide is only acquired in one way, and that's by CO2 diffusion from the outside air through stomatal pores into the photosynthetic tissues. Clicker question. As CO2 concentrations decreased, stomates would need to do what in order to sustain photosynthesis? Open the stomates more? Close the stomates more? Neither open nor close. The answer is A. They would need to open more to allow a faster rate of CO2 diffusion into the photosynthetic cells. Now, this expansion of angiosperms, coincident with the decrease in CO2 concentration, is associated with changes in stomatal density. What we see is that plants went from having a leaf surface, epidermal surface like this, where you have epidermal cells and an occasional pair of guard cells or stomata, to a condition like this, where there's an increase in stomatal density. So as CO2 concentrations went down, stomatal concentrations increased. Well, we can get an idea of how this happened, document it, by looking at fossil leaves, such as the fossil ginkgo, or at modern leaves, such as this modern ginkgo. And if you've never seen a ginkgo before, they are a beautiful deciduous gymnosperm, and there's one located due south of the park building, or immediately due north of Skaggs Biology. So what do we see when we look at stomatal impressions on the surfaces of fossil and modern ginkgos? What we see is a change in stomatal density, known as a change in the stomatal index. It's the number of stomata divided by the total number of stomata and epidermal cells on the surface, times 100. So it's the number of stomata times 100 divided by the number of stomata and epidermal cells. When we look at those stomatal indices, what we see is that today, in a low CO2 world, the stomatal index is about 8.5. We can do experiments of raising plants at higher CO2, and then we find that under higher CO2, the number of stomata goes down, so the stomatal index decreases. However, if we go all the way back to the Triassic and Jurassic, when these fossils were collected, the stomatal indices are much lower, between 5 and 6, 5 and 7. This is taken as evidence that in the Triassic and Jurassic, the stomatal density was much lower, meaning that CO2 concentrations were higher. As the CO2 concentration goes down and more stomata appear, that results in a higher transpiration rate in order to allow greater diffusion of CO2 into the photosynthetic cells. So as the stomata increase, transpiration increases, and to sustain that high transpiration demand, plants needed a more efficient water conducting system. And it turns out that the xylem cells in angiosperms are a much more efficient way to conduct water. Vessel elements are essentially straws on top of each other and nearly open-ended at either end, whereas the xylem and gymnosperms are a series of tracheids, as we learned in the last lecture, which have closed ends. And as a result, water has to go in and out in a circuitous way in the xylem of gymnosperms, but can travel a more direct way in vessel elements of angiosperms. So just to reiterate, the evolutionary advantage of angiosperms is that the vessel element or vessel member allows for faster water transport. Not only are they open-ended, but they're wider. Whereas in the tracheid, we have closed-end and smaller cells, resulting in a lower water flux through the xylem cells of gymnosperms. So as the environment has a decrease in CO2 concentration and stomata open more to allow CO2 diffusion in for photosynthesis, xylem structure of angiosperms with vessel members are a more efficient, more competitive mechanism than we find in the closed-end tracheids of gymnosperms.